This meeting is being recorded.
Hello everyone. Good morning. So I came back with the other classes. I'd like to explain you about cell cycle and cell division previously. I'm continuing that class, cell cycle. The topic is regulation of cell cycle. How cell cycle is regulated. Cell cycle is regulated by two factors. One is external factor and another one is internal factors. So these external and internal factors are signals which come from some from environment and some from the inside of the cell. So you can see here in the picture external signals and internal signals. Here external signals, the growth factor, the external signal, so which will come and stimulate cell cycle to undergo or to stop. And when you see on the right side internal signals, so it is mostly dependent on the kinases, protein kinases, like cyclin dependent kinases, CDKs. Okay, so these protein kinases will regulate the cell cycle. External environmental signals you can see in, for example, Saccharomyces cerevisiae. The two conditions. The two conditions are there for the cell to enter into cell cycle or to not to enter into cell cycle. So here, if it reaches certain amount of size, then it will undergo cell division. Next, unavailability of nutrients. If the nutrients are not sufficient, then also the cell cycle will not complete. So internal signals, I told you, protein kinases, which are the most important regulation of cell cycle. So you can see external and internal signals, how they are activating cell cycle. Next, moving on to yeast cell body. If we see the example Saccharomyces cerevisiae, yeast cells, there is control point that is called as start. And yeast, when we see in eukaryotic organisms, so it is also having same like start that is called as restriction point. So start is the decision point for cell to enter into the cell cycle phases such as G1 to S and S to M phase. Start is present in Saccharomyces cerevisiae, which is resembling in eukaryotic cells, which is having restriction point. Here, start is the decision point. I'm telling that start is the decision point. When to the cell to reach from G1 to S and S to M phase. So you can see the picture here of yeast cell body. So this is for your understanding how yeast cells are body. Just for your understanding, I gave this picture. Next, coming to the internal cell cycle regulators, protein kinases. So the first one, which was discovered in frog oocytes, was MPF. MPF means maturation promoting factor. So this was first discovered in frog oocytes. The scientists have observed it and discovered it. They have taken frog oocytes and injected them with cytoplasm from mature cells into immature ones. So this cytoplasmic material which caused immature ones to mature ones, triggering mitosis. So this led to the realization that diffusible cytoplasmic factor controls mitotic entry. And they found that that is named as maturation promoting factor MPF. So it is very critical regulator of the cell cycle that drives the transition from G2 to M phase. So it may be mitosis or meiosis. So it's a molecular switch that ensures cells only enter division when conditions are right. So this is about MPF. What is MPF means? Maturation promoting factor, which was discovered in the frog oocytes. The scientists have taken frog oocytes, which are not matured, and they have injected with already matured frog oocyte cytoplasm, and they have seen that immature oocytes, when after injecting cytoplasm, they entered into mitosis. So how they have found that some factor is there which is regulating the cycle. Next, when we see the MPF protein complex structure, so it is containing two components. One is cyclin B, which is in golden yellow color, and CDK1 green color. You can see in the right side picture. So these both together will form MPF. PF consists of two proteins, cyclin B and CDK1. So what is cyclin B? Cyclin B is a very important molecule which is acting as a regulatory subunit. Regulatory subunit. And CDK1 is cyclin dependent kinase 1. So it is catalytic subunit. So what are the functions of these things? Regulatory subunit cyclin B, the levels of these are increased during G2 phase and fall after mitosis. When you see CDK, it's a catalytic subunit which phosphorylates targeted proteins to initiate mitosis. Both are together combined, and they will act in cell cycle regulation. Cell cycle regulation. And we see the next slide how MPF works. So you can see on the right side the picture maturation promoting factor, how it works. So it is having two states, inactive state and activation state. In inactive state, CDK1 is present but inhibited by phosphorylation via 1 kinase. See picture here. So V1 is another kinase. So the cyclin B and CDK already attached. It's called MPF, which is activated or inhibited by V1. If V1 is there, inhibit or inactivate the cyclin B and CDK complex. So like that, if V1 is removed, then it will undergo activation. Cyclin B accumulates during G2 phase. Mainly it is accumulated in the G2 phase. So how it is activated? You can see here CDC25 phosphatase enzyme removes inhibitory phosphate from CDK1. So next, cyclin B binds CDK1 and MPF becomes active. So how the MPF works is like this. So you can see in the picture, cyclin B and CDK there. So they are not having any phosphate group here. Okay. CDC25 phosphatase removes inhibitory phosphate from CDK1, and cyclin B binds to CDK1, and then it becomes active. So next slide, what is the function? What are the functions of MPF? MPF phosphorylates proteins that break down the nuclear envelope. And after nuclear envelope is broken down, the, then the chromosomes are condensed, and they'll start assembling the mitotic spindle. So these all are regulated by the MPF, condensing chromosomes and assembling mitotic spindle, which will further can be understood in mitosis. Next, coming to deactivation. Cyclin B is degraded by ubiquitin-mediated proteolysis. So activated, what is the function is we have seen. Next, deactivation means how it is deactivated. This cyclin B is degraded by ubiquitin-mediated proteolysis. So here CDK1 becomes inactive and mitosis ends. In this way, MPF phosphorylates and deactivates the cell cycle regulation. Next, what are the some checkpoints of cell cycle? So in a cell, we have DNA damage checkpoints. If DNA is got damaged, it will not further undergo cell division. If it is continuously enters into damaged DNA enters into new cells, it will be very hazardous to the cell. So that's why in cell cycle regulation, DNA damage checkpoints they they play a very critical role in maintaining the integrity of the genome by arresting cell cycle progression. Arresting cell cycle progression. See, we have the checkpoints in G1 as G2. This will allow time for the damage to be repaired before DNA replication of cell division proceeds. G1, S, and G2 are the places where the checkpoints means they'll check for the damaged DNA is repaired or not. After the repair only, the cell division proceeds. Cell cycle arrest. Talking about cell cycle is arrested, the DNA is damaged. It is mediated by two protein kinases. They are ATM and ATR. ATM means ataxia telangiectasia mutated. ATR means ataxia telangiectasia and the RAD3 of the ATM and ATR stands for these names. These protein complexes recognize damaged or unreplicated DNA. So they will, these ATM and ATR protein complexes recognize damaged or unreplicated DNA. And they will be stopping the cell cycle. They will be arresting the cell cycle. You can see in the picture here. So how they are activated, ATM and ATR. So you can see in the picture, double-stranded DNA got breaks. So then it will stimulate activation of ATM. So ATM is activated by double-strand DNA breaks. ATR is activated by single-stranded or unreplicated DNA. If the DNA is not replicated, it's also very dangerous to the cell. The next generation cell will not get complete DNA. Okay. Next, once activated by DNA damage by double-stranded breaks or single-stranded breaks or unreplicated DNA, ATM and ATR phosphorylate and activate the checkpoint kinase. So you can see the picture here, ATM and ATR phosphorylate CHK1 and CHK. To our ATM and ATR, they are activating CHK1 and CHK2. What are these CHK1 and CHK2 kinases? They will do cell cycle arrest by further phosphorylating or inducing the degradation of CDC25. Then it will lead to the cell cycle arrest. Okay, next. CDC25 phosphatase are required to activate CDK1 and CDK2 by removing inhibitory phosphorylations in cell cycle progression. See further this CDC25 phosphatase which will activate CDK1 and CDK2 and remove the inhibitory phosphorylations and they'll arrest the cell cycle. See, DNA damage leads to inhibitions of both CDK2 and CDK1, resulting in cell cycle arrest.